Yeah, we can start.
Just a minute, ma'am. Yes, ma'am, you can start.
Good evening. Welcome to our exam mastery live classes. So today we'll be talking about biological oxidation, a very important topic. So introducing you to the series. This series is based on rapid revision of all the important topics. We'll be covering all the frequently asked and high yield questions in this series, which is going to be very beneficial for your approaching university exams. Then in today's class, we'll be taking long essay type questions as well as short essay questions. So we'll be talking about the various components of electron transport chain, followed by uncouplers and inhibitors of the electron transport chain. Then what is oxidative phosphorylation? We'll be discussing about that. Then in the short answer types, we'll be talking about the different components, each of them individually. Then a neatly labeled diagram of the electron transport chain that in itself is very explanatory. So even if you're running out of time or you are unable to not write much in the exam, even if you draw a correct diagram, that's going to fetch you marks. Then various inhibitors and the site at which they are acting and their mechanism of action. Then also we'll be discussing about the single electron carrier components of the respiratory chain, and followed by what does oxidative phosphorylation means and how does inhibitors and uncouplers are acting. Okay. Then we'll also be talking about high energy phosphate compounds and PO ratio and oxidative phosphorylation mechanism, and the chemiosmotic theory will also be discussed. Then introducing you to the core features of Auricle. Here at Auricle, we have live classes to elevate your learning experience. Then we also have recorded lectures so that you're able to assess your subjects anytime, anywhere as per your convenience. Then we also provide you with notes based on the latest CBME syllabus. So we have the booklets available now. So this is for biochemistry. Two parts are there, 1 and 2. This is a very precise, bulleted out points, colorful. So if you want them, you can anytime feel free to contact us and purchase the package. Then we also have question banks where we have different MCQs so that you're able to assess your knowledge based on those MCQs. Then we have 3 different subscription plans at Auricle. The basic, which is for 3 months and comes at 15,000, followed by the standard plan, which is for 6 months and comes at 30,000, and then the mentorship plan, which is for 12 months and will cost you 50,000. Then you can get additional 20% off by using my promo code as displayed here at the bottom of the screen. In the mentorship plan, we provide you with personalized guidance and notes. Okay, so with this introduction, let's begin with today's class. So electron transport chain. Before I begin with electron transport chain, let me give you a little introduction. Okay, so an introduction is very important so that you have an idea these questions, why is the cycle taking place, when, where, these type of curious questions, they are going to actually make your background or your base very strong. Okay, so you're able to kind of integrate the various metabolic processes that are taking place. So know that in our body, living cells, they require energy for various processes to take place. Okay, now where is this energy coming from? This energy is coming from oxidation of organic compounds. Okay, so your living cells, they're going to oxidize organic compounds, derive energy from it. Now, some organisms, they are able to make their own organic compounds, like your plants, they're able to photosynthesize their own organic compounds. But like humans and other animals, which cannot synthesize their own organic compounds, they're going to take these organic compounds as food through diet. Okay, so you're consuming food which has these different kind of organic compounds like your carbohydrates, your proteins and fats. Okay, so these are all the respiratory substrates. They're going to undergo respiration. Now, what is respiration? Respiration is a metabolic process where your organic compounds, these respiratory substrate that I mentioned, most importantly, your carbohydrate, glucose. Okay, it is going to get oxidized, and during the oxidation of these organic compounds, energy is going to be released, which is going to be used to generate ATP. Okay, now this respiration of organic compounds, it can be of two types: either anaerobic respiration, which is oxygen independent, or aerobic respiration, which is oxygen dependent. So in aerobic respiration, your molecular oxygen is going to act as the final acceptor of electron. Your organic substrate is going to be completely oxidized to carbon dioxide and water in the presence of oxygen. Okay, now, this aerobic respiration, it involves three important processes: your glycolysis, which we've already discussed, followed by TCA, and then comes your biological oxidation, which is today's topic. Okay, so what have I explained? That you had your food. Your food is going to have different types of organic compounds which are going to get oxidized, okay, through processes like glycolysis, TCA. Ultimately, what is going to happen? Your electrons from these substances, organic compounds that have been oxidized, they are the energy that is obtained from them. It is retained in reduced coenzymes in the form of NADH and FADH2. And then this NADH, FADH2, it is going to give these electrons ultimately to oxygen through a series of complexes, electron carriers, and then oxygen is going to finally accept the electron and water is going to be produced. Okay, oxygen is going to get reduced to water. Okay, so this is the flow of electrons from these reduced coenzymes that are produced during glycolysis and TCA. We're going to study this flow of electrons through the various complexes today. Okay, and this is what is going to constitute your electron transport chain. And then through these flow of electrons, how the energy that is released, that is used to produce ATP, to generate ATP. Okay, so that so your phosphorylation, that is generation of ATP, in coupling with your electron transport chain or flow of electrons, we're going to study that. So that is today's concept or today's topic. That is electron transport chain or biological oxidation. Okay, so with that introduction, let's begin. So let me introduce you to the concept. To on what concept this flow of electron and this biological oxidation or electron transport chain is based. So talking about the concept. Okay, so your electron transport chain, it is a series of redox couples arranged in ascending order of redox potential. Okay, so what is an electron transport chain? If you define it, it is a series of redox couples arranged in ascending order of their redox potential. Now, there are two terms here, that is redox couple and redox potential. Now, what do you understand by redox couples? So, your redox couple, they are those compounds that can exist both in oxidized as well as in reduced state. Okay, so they form your redox couple. Example being like your NAD plus and your NADH. Okay, so NADH is the reduced form and NAD plus is its oxidized state. Okay, similarly your FAD and FADH2. So FAD is the oxidized state, FADH2 is the reduced state. So what is electron transport chain? You have a series, no, not one, but you have multiple such redox couples which are arranged in increasing order of the redox potential. What do you mean by redox potential? Redox potential is the ability to gain electron. Ability to gain electrons. Okay, so more is the redox potential, more is the ability to gain electrons. Okay, How? For example, let's suppose there are two redox couple A and B, and the redox potential of A is less than the redox potential of B. Okay. Now, as per what I've explained, if the redox potential of A is less than redox potential of B, that means B has a greater tendency or more ability to gain electrons. So electrons from this redox couple A, they are going to jump to B. Okay, and this is exactly the concept of electron transport chain, that in electron transport chain you have various redox couples which are arranged in ascending order of their redox potential, or in other words you can say in ascending order of their ability to gain electrons. Okay, so you have complex 1, its ability to gain electron is less compared to its the next complex in the series. Okay, so therefore the electrons are therefore going to jump from one complex to the other in the electron transport chain. Okay, and also when these electrons jump from a lower redox potential to a higher potential, it is going to be an exergonic reaction. That is, free energy is liberated in this process. Okay, while the electrons are jumping from one complex to other, free energy is being liberated, and this free energy that is being liberated, it is used to pump protons from the matrix into the intermembrane space. We will see how. So that was about the definition, or you can say the concept of electron transport chain. So the various reducing equivalents that were generated via different pathways, like your glycolysis, you had carbohydrates, glucose in your body, which is broken down to pyruvate, and through the link reaction it is forming acetyl CoA. Then you have fatty acids which break down and give acetyl CoA via beta oxidation. Then you have glucogenic amino acids. Okay, they are again going to give acetyl CoA. So everything is ultimately converging into your proteins, whether be proteins, carbohydrates, or fat, everything is converging at acetyl CoA. And then this acetyl CoA, it is going to enter TCA, where you're reducing equivalents, that is NADH and FADH2 are produced. These NADH and FADH2, they are then going to enter the electron transport chain. Okay, so if I simplify this for you, what is happening? You have your carbohydrates, your proteins, and your lipids. Okay, they are all ultimately converging at acetyl CoA. They're all forming acetyl CoA. Then this acetyl CoA is going to enter TCA. In the TCA, your NADH and FADH2 are being formed. Those are your reducing equivalents that are formed. Now this NADH and FADH2, it is going to enter the electron transport chain. Okay. Now what happens in the electron transport chain? So NADH is going to give its electron and get oxidized to NAD plus. And your FADH2 is going to get oxidized to FAD plus. Whereas at the same time, what is happening? From the ADP, ATP is being synthesized. So you see, on one end there is oxidation happening, and on the other end there is also phosphorylation taking place. So this is known as oxidative phosphorylation. Okay, so there is oxidation of reducing equivalents here. And also this is coupled with phosphorylation, that is generation of ATP. Okay, so we say oxidation is coupled with phosphorylation. Okay, therefore electron transport chain is oxidative phosphorylation. Okay, so we're getting ATP from reducing equivalents which were generated from food. So you get ATP from reducing equivalents generated from food. Okay. Now let's have a overview of electron transport chain before we begin. Okay. So what is happening? Your NADH is going to come, give its electron to complex 1. Okay. Your FADH2 is going to come and give its electron to complex 2. Both complex 1 and complex 2, they are going to give their electron to coenzyme Q. Now this coenzyme Q will give its electron to complex 3. From the complex 3, it is going to go to cytochrome C, and the cytochrome C is going to transfer the electrons to complex 4. And then from the complex 4, the electrons are going to be received by water. Oxygen is going to be received by oxygen, and oxygen is going to be reduced to water. Okay, so this is an overview. Get this chart and concept very clear so that we can proceed with the individual complexes and you do not mess up things. Okay, NADH coming and giving its electron to complex 1. FADH2, it is going to give its electron to coenzyme Q via complex 2. So you see your complex 1 and complex 2, they are not in series. Okay, both complex 1 and complex 2, they are going to give the electron to coenzyme Q. Then from coenzyme Q, the electron is going to be taken to cytochrome C via complex 3, and then the cytochrome C is going to give the electron to complex 4, and ultimately from complex 4 the electron is going to be received by oxygen. Okay, so this was the overview of electron transport chain. Now let's talk about the components of electron transport chain. So your electron transport chain, it is present in inner mitochondrial membrane. I'm writing that in short as IMM. Okay. So your what are the components of electron transport chain? It consists of fixed 5 multi-protein complexes. That is complex 1, complex 2, complex 3, complex 4, and complex 5. Okay. And it consists of 2 mobile carriers. That is your coenzyme Q and cytochrome C. So we've already seen here in this diagram how your coenzyme Q here and cytochrome C, these are the mobile carriers. Your coenzyme Q is going to go to complex 1 and complex 2, take the electrons from them and going to give to complex 3. Similarly, cytochrome C is going to go to complex 3, take the electrons from complex 3, going to give it to complex 4. So your coenzyme Q and cytochrome C, these are mobile carriers, and rest your complex 1, 2, 3, 4, and 5, they are all fixed in the inner mitochondrial membrane. Okay. Now we're going to talk about the individual complexes one by one. So taking up complex 1. Okay. So your complex 1, it is a large multi-protein complex having multiple subunits. It is fixed, as I've already told you. Okay, and in complex 1, what is happening? Reducing equivalence from NADH, they are going to give this electron. So NADH are the electron donors. And what is happening? Your NADH, after giving its electron, it is being converted to NAD+. So your NADH is going to come, give its electron and gets converted to NAD+. Now what happens from this? Your complex 1, the electron is going to go to coenzyme Q. So you see NADH came, this was complex 1, gave its electron to complex 1, and from complex 1, the electron was taken by coenzyme Q. So what is happening? Your NADH was getting oxidized to NAD+. Okay, so your NADH is getting oxidized. because it is losing its electrons. So loss of electrons is taking place. Loss of electrons is oxidation. So your NADH is getting oxidized, and your coenzyme Q, it is gaining electrons. Okay, coenzyme Q, there is gain of electrons, and gain of electrons is reduction. So we say coenzyme Q is getting reduced. So your NADH gave its electron to complex 1, and from complex 1 it was taken by coenzyme Q. So NADH is getting oxidized, and your coenzyme Q, by gaining electrons, it is getting reduced. Therefore, this complex 1, it is known as NADHQ oxidoreductase. And it is also known as NADH dehydrogenase. And this complex 1, it is a flavoprotein. Okay, this NADH dehydrogenase or complex 1, it is a flavoprotein. And what are flavoproteins? Flavoproteins are those proteins which have FMN or FAD as prosthetic groups. Okay, so in this complex 1 or NADH dehydrogenase, also known as NADH dehydrogenase, also known as NADHQ oxidoreductase, I've explained why it is known so as NADH oxidoreductase. It is a flavoprotein. So that means it is either having FMN or FAD. So here in complex 1, you have FMN. So talking about the components of complex 1. So components of complex 1, it has FMN, one molecule of FMN, and 6 to 7 iron sulfur clusters. Okay, so that was about complex 1. I hope it's clear. Very simple. Now moving on to complex 2. Okay. Now okay, before I talk about complex 2, let me tell you a little bit briefly about coenzyme Q. So your coenzyme Q, it is lipid. It is the only non-protein part of the electron transport chain. So always remember, it is the only non-protein component of electron transport chain. It is the only non-protein component of electron transport chain. It is lipid soluble, okay? And it is basically, it is a lipid soluble benzoquinone with having numerous numbers of isoprene unit, okay? Therefore, it is also known as Q10 because it has 10 isoprene units, okay? And it is a mobile carrier. Okay, so that was briefly about your coenzyme Q. It is taking electrons from complex 1. It also takes electrons from complex 2 and give it to complex 3. Okay, so that was about complex 1, coenzyme Q. Now moving to complex 2. So your complex 2, it is a part of the enzyme succinate dehydrogenase. Okay, succinate dehydrogenase, and I'm sure you all are very familiar with this enzyme. The succinate dehydrogenase, you studied this in TCA, okay? So your succinate forms fumarate, and this was taking place in TCA, and the enzyme was your succinate dehydrogenase. So what happens when from succinate fumarate is being formed? 2 electrons are released, and these electrons which are released, they are first taken up by FAD, and what is formed? FADH2 is formed. Okay, now from FADH2 these electrons are given to iron sulfur clusters that are present in complex 2, and then they are taken by coenzyme Q. Okay, so this is the flow of electrons in complex 2. So what is happening in complex 2? Your electrons are being given from succinate. Okay, so complex 2 is receiving electrons from where? It is receiving electrons from succinate. Okay, and ultimately they are being given to coenzyme Q. And what is the flow of electrons from succinate? The 2 electrons that are released, they are taken up by FAD, which is a component of your complex 2, and then it goes to iron sulfur cluster, again a component of complex 2, and then ultimately to coenzyme Q. So talking about the components of complex 2, so already mentioned FAD. So your complex 2 is also a flavoprotein and then iron sulfur clusters. Okay, so these are the components of complex 2, and in complex 2 what is being oxidized? Your succinate, because electrons are being lost from succinate, so your succinate is being oxidized. Okay, and they are being gained by coenzyme Q. So your coenzyme Q is being reduced. So therefore, this complex 2, it is known as succinate Q oxidoreductase. Succinate, known as succinate Q oxidoreductase. Okay, or succinate Q reductase is another name for it. Succinate Q reductase. So we've studied about complex 2, that how from succinate, when your fumarate is being formed, your enzyme succinate dehydrogenase, it is a part of your complex 2. So the electrons from succinate, they are via complex 2 being taken up by coenzyme Q. So succinate being oxidized and coenzyme Q being reduced. And talking about the components of complex 2, they are your FAD and iron sulfur complex. The flow of electrons being that the 2 electrons that are released from succinate, they are first taken up by FAD, then by iron sulfur complex, and then by coenzyme Q. Okay, now there are 3 major enzyme systems that transfer the electrons directly to coenzyme Q. You see complex 1 here is bypassed. So complex 1 is bypassed. The electrons are directly transferred to coenzyme Q, and 3 enzyme systems are doing this. One we've already taken up, that is your succinate dehydrogenase. Then you have fatty acyl CoA dehydrogenase, and the third one being your mitochondrial glycerol 3-phosphate dehydrogenase. Okay, this we'll study at the end of the lecture when I take up glycerol 3-phosphate shuttle. Okay, so these 3 enzyme systems, they are giving the electrons directly to coenzyme Q. Complex 1 is bypassed. So now your coenzyme Q, it has electrons from complex 1 and complex 2. Now these electrons from complex 1 and complex 2 that are taken up by coenzyme Q, they are going to be given to complex 3. So let's talk about complex 3 now. Complex 3. So the electrons from coenzyme Q will jump to complex 3. So this was your, okay, so this was your coenzyme Q. It has received electrons. Now it is going to give electrons to complex 3. So electrons from coenzyme Q, they are going to be given to complex 3. Now from complex 3, the electrons will be taken up by the next mobile carrier, that is cytochrome C. Okay, so basically electrons from coenzyme Q are being lost. So your coenzyme Q is getting oxidized and they are being received by cytochrome C. So your cytochrome C is getting reduced, since it gains electrons and electron is lost from here. Okay, now what happens? As your coenzyme Q is being oxidized and cytochrome C is being reduced, therefore this complex 3, it is known as Q cytochrome C oxidoreductase. Okay, so that is the name of your third complex, Q cytochrome C oxidoreductase. It is also cytochrome B C1 complex. Okay, now talking about the components of complex 3. So it is cytochrome B, cytochrome C1 and rescue iron sulfur complex. Okay, so these are your components of complex 3, that is cytochrome B, cytochrome C1 and rescue iron sulfur complex. Therefore, this complex 3, it is also known as cytochrome B C1 complex. And why is it known as Q cytochrome C oxidoreductase? Because from coenzyme Q, the electrons are being given to cytochrome C via complex 3. Okay, so we've talking about the components of complex 3. What are it? Cytochrome B, C1 and rescue iron sulfur complex. We've also taken about the different names, cytochrome BC1 complex, or also known as Q cytochrome C oxidoreductase. Now moving on to complex 4. So your complex 4, it is going to receive electrons from cytochrome C and then this electron is going to be given to oxygen. So talking about complex 4. So complex 4 is receiving electrons from cytochrome C. So electrons from cytochrome C is given to complex 4 and then from complex 4 the electrons are given to oxygen. Okay, so your cytochrome C, as you can see here, is getting oxidized because it is giving its electrons and the electrons are being received by oxygen. Oxygen is the final or the terminal electron acceptors. So your oxygen is getting reduced. Oxygen is getting reduced to water. Okay, so your oxygen is getting reduced to water, your cytochrome C is getting oxidized, and therefore this complex 4 is also known as cytochrome C oxidase. Cytochrome C oxidase. Also, always remember that your oxygen is the final electron acceptor, and your oxygen, in order for it to get completely reduced to water, it should receive 2 electrons. Okay, on receiving 1 electron, it forms superoxide. Okay, so for oxygen to be completely reduced to water, it should always receive 2 electrons. And it is an irreversible complex. And talking about the components of this complex 4, so components of complex 4, there are your heme AA3, which is also known as cytochrome AA3. Okay, and it also has 2 copper centers, copper A, copper B center. So what are the components of complex 4? It has 2 heme groups. 2 heme groups just mentioned. Heme A and heme A3. What are these? Are the 2 heme groups? Okay, heme A and heme A3. So heme A is also known as cytochrome A and heme A3 is also known as cytochrome A3. And both heme A and heme A3, they're having distinct properties. And also I told you they have 2 copper centers. 2 copper centers. What are they? Copper A and copper B centers. So your copper A, it contains two copper ions, and your copper B center, it contains one copper ion. And I hope you all know what is cytochrome. Cytochromes, these are heme proteins, okay? So that was about complex 4. That complex 4, it is receiving electrons from cytochrome C and giving it to oxygen. Oxygen is the final acceptor of the electrons, and oxygen is getting completely reduced to water. So complex 4, it is having two heme groups, heme A and heme A3, and it is also having two copper centers, copper A and copper B. So talking about the flow of electrons through complex A, if I discuss that, so your cytochrome C was give its electron first to copper A center, then it is going to be given to cytochrome A, which is also known as heme A, then it is going to cytochrome B, copper B center, cytochrome A3, also known as heme A3, and then finally taken up by oxygen. Okay, so this is the flow of electrons. Okay, so your oxygen being the final electron acceptor. Okay, so with this, we've taken all the individual complexes. So this is what your electron transport chain looks like. I'm going to explain this diagram. You see, from NADH, your electrons were given to complex 1 via the FMN and iron sulfur clusters, which are the components of your complex 1. And in doing so, the free energy that was being liberated, that was used to pump 4 hydrogen atoms from the matrix into the intermembrane space. Okay, now your complex 2 comes in picture. So here, what are you getting? From FADH2, from like mitochondrial glycerol 3-phosphate or through succinate dehydrogenase, they are going to give their electrons to FAD, and then it is going to form FADH2. Then that FADH2 is going to give its electron to FAD, and then to iron sulfur clusters, and then to coenzyme Q. So finally your coenzyme Q is receiving electrons from complex 2 as well as from complex 1. Then coenzyme Q is going to give its electron to complex 3, and complex 3 is also acting as a proton pump, and it pumps 4 protons into the intermembrane space for a pair of electrons transferred. Okay, now from complex 3, the electrons are going to be taken up by cytochrome C. Cytochrome C is going to give the electrons to complex 4, and complex 4, they are having two copper centers, two heme centers. Okay, from complex 4, the electrons are then given to oxygen, oxygen being the final acceptor of electrons, and it is going to get reduced to water. Okay, and complex 4 is also acting as a proton pump, and it pumps 2 hydrogen into the intermembrane space. Okay, and always remember your complex 2 does not act as a proton pump. Okay, so here it was from succinate. The complex 2 is again giving it to coenzyme Q, and then it is being received by complex 3. And complex 3, we know through cytochrome, it is going to give it to complex 4. So this is the cycle for the complex 2, and here this was from NADH. Okay, so I hope it's clear, the electron transport chain. Now let's talk about phosphorylation. So this was the complexes that were involved in oxidation. Now coming on to the phosphorylation part of biological oxidation or electron transport chain. Okay, so coming on to complex 5 now. So your complex 5, it is involved in phosphorylation or ATP synthesis. Okay, this complex 5, it is also known as ATP synthase. So your ATP synthesis complex 5, it is the smallest molecular motor present in the body. Smallest molecular motor present in our body. Now talking about the components of complex 5. So your complex 5, it consists of two subcomplexes. Two subcomplexes. What are they? It consists of F0 subcomplex and F1 subcomplex. Okay, so talking about F0. So F0, it is made up of 10 C disc proteins. Okay, and this F0 is acting as a proton channel. Through these F nodes, your proton that were being pumped out into the intermembrane space, they are again going to come back into the matrix. So remember that F node is acting as the proton channel. It is hydrophobic. It is made up of 10 C disc proteins and it spans the inner mitochondrial membrane. So it is present in the inner mitochondrial membrane. So talking about F1, so F1 is tightly bound to F0 and protrudes into the matrix. Okay, so your F1 is protruding into the matrix and it is connected to F1 via its gamma subunit. We'll talk about this. I'll show you the picture, it will become more clear. And your F1 is made up of 9 subunits. Okay, what are those subunits? 3 alpha, 3 beta, and 1 gamma, delta, and epsilon. Okay. So if you see the diagram, it's going to become more clear here. So this is your complex 5. Complex 5, also known as ATP synthase. And this complex 5, this is involved in ATP synthesis. The rest of the complexes that we've studied from 1 to 4, they were involved in electron flow. Now this one is involved in ATP synthesis. Okay. So I was talking about complex 5. I said it has two subunits, F0 and F1. So this is your F0 and this is your F1. Your F0, it is consisting of 10. You can see these C disc proteins. So these are the part of your F0. They are present in the inner mitochondrial membrane and they are acting as proton pumps. So that was about F0. Then I said your F1 is protruding into the matrix. So you can see this is how it is protruding into the matrix on the inside. This is the matrix side. Okay, so F0 is protruding into the matrix and it is connected to, sorry, F1 is protruding into the matrix and it is connected to F0 via the gamma subunit. Okay, now your F1 is having 3 alpha, 3 beta, 1 gamma, 1 delta, and it also has 1 epsilon. The epsilon is also present here on the axis. Okay, so it has 3 beta, which are going to be the ATP synthesizing subunits. We will study this in chemiosmotic theory. It has 3 alpha, 1 gamma, 1 delta, and 1 epsilon. So this is your delta, this is your gamma, and this is your epsilon. Okay. Now the gamma subunit, it forms the bent axle. As you can see, gamma is forming the axle, which is joining the F0 to the F1. And also there is epsilon present on it. Okay. So your gamma is going to be the rotatory subunit. What happens is the entire of the ATP synthase is not rotating. So F0 I said is going to act as a proton channel. So when protons are going to pass through the F0, this is going to make your C disc proteins and the gamma to rotate. Okay. So basically, what are your rotors being formed from? Your gamma, which is forming the bent axle along with epsilon. So your gamma and epsilon, which are present in the axle, and your C rings of F0. Okay. So these, they are going to form the rotor. Okay, and rotation movement is imparted to the rotor by passage of protons. Okay, so when protons are going to pass through the F0, which I said is going to act as a proton channel, then what is going to happen? This rotor, it is going to rotate. So only the rotor is going to rotate, not the entire ATP synthase complex. Okay, so entire ATP synthase complex is not rotating. Only your axial, which is formed by gamma and epsilon, and the C rings of the F0. They are the rotator rotatory unit or the rotor, and the rest of the ATP synthase is forming the stationary unit. Okay, so that was about the structure of complex 5. Now let's talk about oxidative phosphorylation and chemiosmotic theory. Okay, so talking about oxidative phosphorylation and chemiosmotic theory. So we have seen how your electrons were jumping from complex 1 to complex 2, 3, 4, and ultimately what was happening, your protons were also being pumped out. Okay, so if we talk about NADH, so from NADH, the electrons were being given to complex 1, and then from complex 1 it was going to coenzyme Q, from coenzyme Q to complex 3, and from complex 3 to cytochrome C, and from cytochrome C to complex 4, and finally from complex 4 it was the electrons were taken up by oxygen, and oxygen was completely reduced to water. So this is the flow of electrons for your NADH. And for FADH2, what was happening was via complex 2, and it was being given to coenzyme Q. Complex 1 was bypassed. Okay, so you see the electrons, they jump from one complex to another. And as they are jumping, free energy is being released. Okay, so jumping of electrons from one complex to another, this releases energy, and this energy that is being released, it is used to pump protons from the matrix into the intermembrane space. So these complexes, they are going to act as proton pumps. So when electrons jump from one complex to another, with it, each of the complex is going to pump protons to the intermembrane space. Now your complex 1, it pumps 4 protons in from the matrix into the intermembrane space. Then your complex 3, it is also pumping 4 protons, and your complex 4, it is pumping 2 protons. Okay, so there are a total of 10 protons being pumped. 10 protons or 10 hydrogen ions per electron pair. Okay, so when electrons are jumping, free energy is being released, which is being trapped to pump protons, in other words, being trapped to generate ATP. Also remember that your complex 2 is not acting as a proton pump. Okay, complex 2 does not act as a proton pump. So from NADH, you are getting 10 protons, and from FADH2, complex 1 is bypassed, complex 2 is not acting as a proton pump. From complex 3 you will get 4 protons, and from complex 4 you will get 2 protons. So you're getting 6 protons from FADH2, and from NADH2 you're getting 10 protons. Okay, now let's talk about the chemiosmotic theory. So what is happening because of this? This is your intermembrane space. Okay, okay, let's suppose this is not your intermembrane space, this is your inner mitochondrial membrane. Okay, this is your inner mitochondrial membrane, this is the matrix site inside the mitochondria, and this is the intermembrane space. Now what happens because of jumping of electrons from one complex to other? I said your complexes, they are going to act as proton pumps. So they are going to pump protons from the matrix to the intermembrane space. So as a result of this pumping, what is going to happen? There's going to be less protons in the matrix side and more protons in the intermembrane space. Okay, so your intermembrane space, there is more positive charge. It is going to be more positive, and your matrix is going to be less positive or negative. Okay, and also it is going to have higher pH because hydrogen ion concentration is low, and here the hydrogen ion concentration is low, so it is going to have lower pH. So the intermembrane space is having more of positive charge, the matrix is having less of positive charge, or it is negative. So potential difference is created due to pumping of protons. Okay, so there is more positive charge accumulating in the intermembrane space, and a potential difference has been created. Now what happens, because of this potential difference, the proton would want to go into the matrix from the intermembrane space. Now F naught subunit is going to act as a proton channel. So the matrix which is less positive as compared to the intermembrane space, so what is going to happen through the F naught channel, which is going to act as a proton pump? Because of this potential difference that is created, the protons are going to move into the matrix through the F naught subunit. I hope it's clear that because of the pumping of electrons from the matrix into the intermembrane space via the complexes, as the electrons were jumping, there was more of positive charge accumulated in the intermembrane space. Now, because of this potential difference that is created due to accumulation of positive charges in the intermembrane space, the protons via the F naught unit, which is going to act as a proton pump, move into the matrix. Okay, now as the protons move through the F naught, what happens? Rotation is going to take place. Okay, so the movement of electrons to F naught, it causes or imparts rotation to the rotor, that is to the gamma subunit and the C proteins. Okay, so as the protons move through the F naught, rotation is going to take place in the gamma subunit along with the C proteins of the F naught, and because of this rotation, what is going to happen? Conformational changes in the beta subunit. Okay, the beta subunit of your F1 are going to take place. And as a result of these conformational changes, your three beta units, they're going to convert ADP into ATP. And this is phosphorylation. Your ATP has been generated. So because of jumping of electrons, that was oxidation. What was happening because of that oxidation? The free energy that was being liberated, that was used to pump protons. And because of the pumping of protons, there was positive charge accumulating in the intermembrane space. Okay, now a potential difference was created. Now because of this, the protons from the intermembrane space, they move through the F naught, which is acting as a proton channel, into the matrix, and because of the movement of protons through the F naught, what is happening? Your rotation is going to take place. Okay, so as your gamma subunit along with the C proteins of the F naught, they rotate, conformational changes take place in the beta subunit, and your ADP is converted into ATP. So if I bullet this out, there was flow of electrons through the various complexes. Okay, flow of electrons to various complexes, which was accompanied by pumping of protons, and as protons were pumped, a potential gradient was created, and because of that potential gradients or potential difference, what happened? Your F naught, it acted as a proton channel, and from the intermembrane space through F naught, your protons, they moved into the matrix, because of which rotation took place in the gamma and the C proteins of the F naught. And because of the rotation, what happened? Conformational changes occurred in the three beta subunits of F1. And because of these conformational changes, your ADP was converted into ATP, and this is known as phosphorylation. And here this flow of electrons, it was known as oxidation. So you see how your oxidation is coupled with phosphorylation, and this is what is known as oxidative phosphorylation. Okay, so you've seen how oxidation is coupled with phosphorylation, and this theory of oxidative phosphorylation is known as chemiosmotic theory, and it was given by Peter Michel. So this is known as chemiosmotic theory, and it was given by Peter mission. Okay, so I hope it's clear. So with this, we come to the end of oxidative phosphorylation or biological oxidation. And I don't think there's anything left to explain. I've explained you in detail, and if you've paid attention, I think that was more than enough. Now, talking about the inhibitors of electron transport chain. So inhibitors can either be inhibitors of the electron transfer. So inhibitors of the electron transfer, they are basically going to inhibit the complexes. Now they can also be inhibitors of oxidative phosphorylation and then uncouplers. So there are three types of inhibitors. So talking about the inhibitors of the complexes. So these inhibitors, they are going to inhibit your electron transfer. So talking about the complex 1 inhibitor. Complex 1, the inhibitors are you rotenone, amobarbital, and pyrisidin A. So your rotenone and amobarbital, they are more or less working in a similar fashion. What happens is they are going to inhibit complex 1. So from NADH via complex 1, the enzyme was going to coenzyme Q. Now this is inhibited. Okay, what is rotenone? Your rotenone, it is a plant product and it inhibits your complex 1. Therefore, your rotenone, it is used as a fish poison or an insecticide because it has been found that in various fishes and in insects, it is strongly inhibiting your complex 1. Amobarbital also inhibits complex 1. Your pyrisidin A, it competes with coenzyme Q. So instead of your electron passing to coenzyme Q, it is given to pyrisidin A. Now talking about the inhibitors of complex 2. Complex 2 inhibitors, they are your malonate, because your malonate, it inhibits succinate dehydrogenase. And then you have carboxyl, and then you also have trienoyl trifluoroacetate. So these are all your inhibitors of complex 2. Then inhibitors of complex 3, they are your British anti-leucite and anti-mycin A. Then inhibitors of complex 4, they are your carbon monoxide, cyanide, hydrogen sulfide, all are gases, okay, and sodium azide. So they are your inhibitors of complex 4. Now talking about inhibitors of oxidative phosphorylation. Those were inhibitors of electron transport. Okay, your electron was not being transported because your complexes were being inhibited. Your flow of electrons was not taking place. Now talking about inhibitors of oxidative phosphorylation. So here oxidation will happen, but it is not coupled with phosphorylation. Okay. So these are going to cause blockage from complex 5 onwards because your complex 5 was involved in phosphorylation. Okay. So that is F naught is going to be blocked. Okay. Here the F naught, the O here is actually denotes oligomycin. Oligomycin. Okay, so the F naught, the O in F naught is actually denoting oligomycin. And what is oligomycin? It is an antibiotic which is inhibiting your F naught. So oligomycin, it inhibits your F naught. And what was F naught doing? It was acting as a proton pump. Okay, so your oxidation is going to happen, but your phosphorylation is not going to take place. And your F1, it is inhibited by orovertin. And also there is this inhibitor known as atractyloside. Now, what is atractyloside inhibiting? Your inner mitochondrial membrane, it is not freely permeable to ATP and ADP. So, the ATP that is synthesized in the matrix, it needs to go out into the cytoplasm, and your ADP needs to enter into the matrix from which ATP can be synthesized. So, for this, there is a transporter in the inner mitochondrial membrane. This is your membrane. There is this transporter, and this is known as ATP, ADP, ATP, ADP transporter, which is known as translocase. Okay, it is an antiporter. So it is pumping into the matrix. It is pumping ADP, and the ATP that is synthesized, that is being moved out. So this ATP ADP translocase or transporter, this is inhibited by atractyloside. So these were the inhibitors, your oligomycin, ariovertin, and your atractyloside. These were the inhibitors of oxidative phosphorylation. Now, what are uncouplers? Uncouplers, they are going to dissipate your proton gradient. Okay, so they're going to actually create holes, form channels, or something like that, through which your protons, rather than moving through the F naught, they're going to move through these holes, and therefore, though your proton gradient is going to be dissipated. Okay, so they allow oxidation of NADH and FADH2 to take place. Okay, and oxygen being reduced. This is going to continue, but they do not permit ATP synthesis. So ATP synthesis is not taking place. And examples of your chemical uncouplers, they are your DNP, that is your 2,4-dinitrophenol, then dinitrocresol, then you have FCCP, that is your fluorocarbonyl cyanide phenyl hydrazine, and you have aspirin and hydrosis. So these are your chemical uncouplers. If I talk about the functioning of DNP, it is similar to CCP. And how does it function? Your DNP or 2,4-dinitrophenol, in its anionic state, it is able to uh, pick up a proton. Okay, so in its anionic state, your DNP, it picks up a proton, and this DNP, it is lipid soluble. Okay, so in its anionic state, it picks up a proton, and since it is lipid soluble, it diffuses across the inner mitochondrial membrane, and in the matrix, it releases the proton. So in this way, it is dissipating the proton gradient, which is very essential for phosphorylation. Okay, and thereby inhibiting ATP synthesis. Now talking about physiological uncouplers. So you have some physiological uncouplers like your thermogenin. Then you have long chain fatty acids, and you have thyroxine. So these are your physiological uncouplers. If I talk about thermogenin, this is important. Thermogenin, it is a protein which is present in the inner mitochondrial membrane of adipocytes. Your bats, that is brown adipose tissues. Okay, they are rich in mitochondria, they have high mitochondria, and in the inner mitochondrial membrane there's this protein known as thermogenin present, and this thermogenin, it provides alternate pathway for protons. So what is going to happen? The energy that is released due to flow of electrons, it is not used to generate ATP, rather it is used to generate heat. Okay, and this actually is responsible for non-shivering thermogenesis in neonates and in hibernating animals. And your neonates, they have fluffy and chubby cheeks, okay, which are rich in brown adipose tissue. And in these brown adipose tissues, you have thermogenin, which is responsible for generating thermogenesis or responsible non-shivering thermogenesis, okay, generating body heat. Since these thermogenin, they are providing an alternate path for the protons. Okay, so therefore your ATP synthesis is not taking place, rather the energy that is released due to flow of electrons, it is used to generate heat. Then you have ionophores. Now what are ionophores? These are lipid soluble compounds that can increase the permeability of the lipid membrane to certain ions. Okay, so your ionophores, they are lipid soluble compounds which increase the permeability of the membrane to different ions. Now they can be of two types: mobile ion carriers, or they can be channel formers. channel formers, your example being gramicidin. So what happens is this is your inner mitochondrial membrane, okay, and this was your F node, and this was your proton channel. The proton was supposed to pass through this F node. Now what happens, because of channel formers, there are going to be ion channels formed for other ions. For example, let's suppose for potassium. So what is going to happen? Potassium is going to move through this channel that is being formed and dissipate the potential gradient that was created because of pumping of protons. So ionophores, they are can be either channel formers or they can be mobile ion carriers. For example, your valinomycin. What does valinomycin do? Your valinomycin, it combines reversibly with potassium ions. Okay, so your valinomycin, it combines with potassium ions and it forms a membrane permeable complex. Now this complex, it is going to diffuse to the inner mitochondrial membrane and thus thereby dissipate the your proton gradient that has been generated, and therefore your ATP synthesis will not take place. Okay, so these were also one kind of uncouplers or inhibitors of your electron transport chain. So with this, we come to the end of the lecture. I hope you